and welcome to today's webinar event, a 16-color flow cytometry panel to monitor checkpoint inhibitor expression and T-cell exhaustion following treatment with the PD-1 inhibitor nivolumab. This is Michael Krieger with Bio-Rad. Thanks for joining us today. With us today is Dr. Angie Green, Senior Scientist with Bio-Rad here in Berkeley. A little bit about Angie. She's been with Bio-Rad for the past 3 years, and before joining Bio-Rad, she was postdoctoral scientist at UC Berkeley School of Public Health and held postdoctoral fellowships at both UC Davis School for Comparative Medicine and the University of Pittsburgh School of Medicine after receiving her PhD there in microbiology and immuno-oncology. Before I turn it over to Angie, I want to let you know that today's webcast is completely interactive, and so we encourage your participation by asking questions. To do so, just press the Q&A button, type your question into the Q&A pane, and press Submit. We'll answer as many questions as possible, and ones we don't have time for, we'll receive a written response after the event. You can also use the Green Resources widget to download more information related to the flow cytometry applications. And every window you currently see, from the slide window to the Q&A window, can be enlarged or collapsed. So if you want to change the look and feel of your console, go right ahead. For this event, I suggest you maximize the slides window or even make it full screen, since there is a lot of detail on Angie's slides. Finally, when we're done, we'd appreciate if you complete our post-webinar survey to let us know how you like this event. You'll see the link when the event completes in the lower right-hand side of the screen. And with that, Angie, over to you.
Thank you, Michael, for the lovely introduction and providing the background for what we'll be discussing today. And thank you to our audience for joining us. If you have any questions, as Michael noted, please make sure you enter them into the Q&A chat box found on the interface. And now on to what we'll be discussing. Today we'll be using a 16-color flow cytometry panel to monitor checkpoint inhibition expression and T-cell exhaustion following treatment with a PD-1 inhibitor. T-cells play a key role in immunosurveillance for cancer, and tumors have developed sophisticated mechanisms to evade these T-cell responses, such as inducing T-cell exhaustion and signaling through checkpoint inhibitors. Immunotherapy has provided mechanisms to reinvigorate tumor-specific T-cell responses and improve clinical outcomes. However, not all patients achieve a long and lasting cure. For effective chemotherapy, it is critical to understand resident T-cell populations, the activation state of the T-cell, and the expression profile of the checkpoint inhibitors. Tumor-specific T-cells eliminate cancer cells via direct and indirect killing mechanisms. Signaling through the TCR, the cytotoxic T-cell can kill tumor cells directly via release of perforin and granzyme, as well as upregulation of Fas ligand to induce cellular death. CD8 toxic T-cells directly kill cancer cells when the T-cell recognizes tumor antigen in the context of MHC class I molecules. Cytotoxic CD4 T-cells can kill cells directly that express tumor antigen in the context of MHC class II. Tumor cells that often express MHC class I but lack MHC class II expression. Thus, CD4 T-cells cannot recognize the tumor cell directly that lacks MHC class II. When cancer cells lack MHC class II, CD4 T-cells are able to play a role via a mechanism of indirect killing. Tumor antigen is secreted by tumor cells and can be processed and presented by an MHC class II antigen-presenting cell, such as a macrophage. The T-cell can then recognize the tumor antigen, bind to the macrophage, resulting in activation of both the CD4 T-cell and the macrophage. The activated macrophage can be then tumoricidal and kill the tumor cell. Chronic T-cell stimulation can lead to T-cell exhaustion, characterized by progressive loss of cellular function and sustained expression of checkpoint inhibitors. Checkpoint inhibitors are cell surface receptors that play an important role in downregulation of immune responses and promotion of self-tolerance by inhibiting T-cell inflammation. Within the tumor environment, tumor cells can bind checkpoint inhibitors such as PD-1 and inhibit tumor-specific T-cell responses. The use of blocking antibodies as a checkpoint inhibitors has shown that T-cell exhaustion is reversible. To date, 5 PD-1, PD-L1 blocking monoclonal antibodies have been approved by the U.S. FDA, and their efficacy has been confirmed in the treatment of several cancers. According to the Cancer Research Institute, there are currently over 1,500 clinical trials testing the popular checkpoint inhibitors. Immune therapy has been effective for people with certain types of cancer, such as melanoma, that are resistant to chemotherapy and radiation therapy. Cancer treatment with immunotherapy offers the possibility of a long-term cancer remission. Immunotherapy can train the immune system to remember cancer cells. The immune memory may result in a longer-lasting remission. When compared to chemotherapy and molecular targeted therapy, immune checkpoint inhibitors have a higher rate of primary resistance that diminishes the overall clinical benefit. The efficacy of monotherapy with PD-1, PD-L1 blocking was rarely greater than 40%. Additionally, after an initial response to PD-1, PD-L1 blockage, most patients acquired resistance that later led to disease progression and relapse. Primary resistance to PD-1 therapy is associated with overexpression of PD-1, PD-L1, and severe T-cell exhaustion. The mechanisms of resistance are complex, dynamic, and interdependent. There are many tumor cell intrinsic and extrinsic factors related to PD-1 blockade resistance. These factors include PD-L1 expression, tumor neoantigen expression and presentation, associated cellular signaling pathways, tumor microenvironment, related immune genes, and epigenetic modifications. In addition to PD-1, overexpression of multiple inhibitory receptors including TIM3, CTLA4, LAG3, and BTLA are associated with inhibition of T-cell function and resistance to PD-1, PD-L1 inhibition therapy. To better understand the complex interplay and expression profile of these checkpoint inhibitors, the 16-color panel was designed. To evaluate the effects of PD-1 inhibitor treatment during chronic T-cell stimulation, an in vitro model of pan-T-cells was used in lieu of clinical samples. T-cells were stimulated with anti-CD3 CD28 beads for 0, 4, and 13 days in the presence and absence of the PD-1 inhibitor nivolumab. Following stimulation, the cellular profiles were analyzed using a fully validated 16-color panel. The 16-color panel was designed to identify T-cell subsets and evaluate the expression of inhibitor receptors within these subsets. In addition to inhibitory molecules, T-cell activation markers relevant to clinical samples, such as CD28 and CD25, were also evaluated. Studies have shown that CD28 is required for PD-1 therapy to kill cancer cells effectively and eliminate chronic viral infection in mice. Lung cancer patients that responded to PD-1 therapy had more CD28 positive T cells, suggesting that CD28 expression may predict treatment response. Finally, T-cell associated cytokine profiles were evaluated. IL-2, TNF, and interferon gamma expression by T cells may influence the tumor microenvironment. These samples were collected using a 5-laser ZE5 Cell Analyzer, which allowed us to use a very small volume of precious sample for our analysis. For analysis of our in vitro model, T cells were identified as CD3 positive cells within the lymphocyte gate. They were further broken down into the CD4 positive and CD8 positive T cell compartments. Within the CD4 T cell compartment, regulatory T cells were identified using CD25 and CD127 low positive cells. Naive, central, effector, and terminally differentiated memory cells were further characterized in both the CD4 and CD8 T cell compartments using CD45RA and CCR7. Over the course of T cell stimulation, the T cells became increasingly differentiated, and underwent progressive loss of key surface molecules. Naive cells lost CD45RA first, and then CCR7, eventually losing CD28 and CD27 expression. The exception of this loss for CD28 and CD27 was the terminally differentiated effector memory CD8 T cells. With continued stimulation, expression of the inhibitory checkpoint markers increased and cytokine production decreased. Cells were first gated on the lymphocyte gate and doublets removed using forward and side scatter parameters. The live cell population was then identified using a VivaFix dye. As you can see, a majority of the sample is CD3 positive, as expected for a pan-T cell sample used in the in vitro model. Within the sample, a distinct population of Tregs can be identified. Tregs are important because checkpoint inhibitors reduce apoptosis in these regulatory T cells, and regulatory T cells play an important role in limiting inflammation and suppressing T cell function. During cancer treatment, PD-1 blockade is used to reverse T cell exhaustion and invigorate T cells specific for cancer. In this in vitro model of T cell exhaustion, the expression profile of several checkpoint inhibitors were evaluated on both CD4 and CD8 T cells. First, we analyzed the expression of PD-1 on both CD4 and CD8 T cells in the presence and absence of the PD-1 inhibitor nivolumab. This analysis was repeated for the additional checkpoint inhibitors TIM3, LAG3, and CTLA4. Both CD4 and CD8 T cells had increased PD-1 expression on the surface by day 4 in the absence of treatment. Interestingly, on day 4, in the presence of PD-1 inhibition, T cells expressed more PD-1 on the surface than the control. CD4 T cells had a twofold increase, and CD8 T cells an almost fourfold increase in the PD-1 expression in the presence of PD-1 inhibition. This suggests that blockade of the PD-1 increases surface expression on these cells. By day 13, expression of PD-1 was higher in the control than the treated within the CD4 compartment, whereas both treated and untreated CD8 T cells expressed similar levels of PD-1. Over time, both TIM3 and PD-1 exhibited the most dramatic changes in expression on both CD4 and CD8 T cells. Within the CD4 T cell compartment, expression of PD-1 and TIM3 were increased in the presence of nivolumab by day 4. TIM3 expression remained elevated on CD8 T cells for both day 4 and day 13. CTLA4 and LAG3 had modest increases in expression by day 4, but expression levels did not appear to be affected by PD-1 inhibition. To assess how PD-1 inhibition may affect T cell subsets, CD4 T cells and CD8 T cells were analyzed using CD45RA and CCR7. The use of these two markers is able to discriminate naive, central memory, effector memory, and terminally differentiated effector memory cells within each T cell subset. When T cells are stimulated without inhibitor, fewer naive CD4 and CD8 T cells were observed. An increase in the central memory phenotype was observed by day 4, and by day 13, the majority of CD4 and CD8 T cells were either a central memory or effector memory phenotype. CD8 T cells had a small percentage of terminally differentiated effector memory cells by day 13. In the presence of the PD-1 inhibitor, CD4 and CD8 T cells appear to have fewer naive cells develop into a memory phenotype at day 4, whereas by day 13, cells treated with the PD-1 inhibitor had fewer naive cells than the non-treated controls in both compartments. These data suggest that PD-1 inhibitor affects phenotype development. Naive and memory cell populations were evaluated for checkpoint inhibition expression in the presence and absence of the PD-1 inhibitor. Within the CD4 T cell compartment, the highest frequency of PD-1 positive cells was observed on day 4 when CD4 T cells were in the presence of the PD-1 inhibitor. We then assessed the frequency of PD-1 expression within the naive, central, and effector memory populations. At day 0, prior to stimulation, a small number of central and effector memory cells expressed PD-1. By day 4, the frequency of PD-1 expressing cells increases most within the central memory compartment, with fewer of the effector memory cells expressing PD-1. By day 13, both central and effector memory cells are producing similar amounts of PD-1. Interestingly, CD8 T cells had a similar PD-1 expression pattern when compared to CD4 T cells. CD8 cells stimulated in the presence of PD-1 inhibitor for 4 days had the highest frequency of PD-1 positive cells. This PD-1 expression was mostly on the central memory compartment, with fewer of the effector memory cells expressing it. This is interesting because patients with effector memory populations that are CD28 positive and PD-1 intermediate phenotype have been shown to have a greater response to PD-1 therapy during immunotherapy. In addition to PD-1, TIM-3 was the second checkpoint inhibitor that showed the most dynamic change in expression over the course of the experiment. The expression of TIM-3 was evaluated in both the CD4 and CD8 T cell subsets. Within the CD4 T cell compartment, cells treated in the presence of PD-1 inhibitor exhibited the highest frequency of TIM-3 positive cells, a majority of which came from the central memory compartment. Very few effector memory cells expressed TIM-3. TIM-3 expression in CD8 T cells increases by day 14 and is maintained through day 13. Treatment with the PD-1 inhibitor slightly increased the frequency of TIM-3 positive cells within the CD8 T cell compartment. Prior to stimulation, naive cells account for the majority of TIM-3 positive cells, whereas central memory cells account for the majority of TIM-3 positive cells following stimulation, regardless of the presence or absence of the PD-1 inhibitor. Cytokines are produced by T cells and not only influence the microenvironment of the immune response, but shape T cell responses. To fully assess T cell functions in our in vitro model of T cell exhaustion, IL-2, TNF, and interferon gamma production were monitored throughout the course of the experiment. We first analyzed cytokine production within the total CD4 and CD8 T cell compartments. As expected, the overall frequency of cytokine-producing cells increased between day 0 and day 4 within both the CD4 and CD8 T cell populations. CD4 and CD8 T cells treated with PD-1 inhibitor had a greater percentage of cells producing IL-2 at day 4 and 13 when compared to the control. Interestingly, the presence of the PD-1 inhibitor did not alter the frequency of TNF or interferon gamma producing cells in either population. CD4 and CD8 T cell subsets were examined to determine the source of cytokine production. CD4 positive T cell central memory had the highest frequency of cytokine production, followed closely by the effector memory population. Examination of the CD8 T cell compartment showed similar data. The highest frequency of cytokine-producing cells was found within the central memory, followed closely by the effector memory. Interestingly, the terminally differentiated effector memory cells had a very low frequency of cytokine production. The frequency of cytokine producing cells within each T cell subtype did not appear to be affected by PD-1 inhibition. Using an in vitro model of T cell exhaustion and PD-1 blockade, we have shown you data from a 16-color panel used to track the dynamic changes in T cell subsets, checkpoint inhibitors, and cytokine expression. Instead of comparing cytokine and surface markers to individual subsets, we have analyzed the data using T-SNE plots. T-SNE analysis reduces multiple dimension data into 2 dimensions. At the same time, data points that were close to one another in the original data remain close in the final T-SNE analysis. For both CD4 and CD8 T cells, T-SNE plots were used to compare checkpoint inhibitor and cytokine expression between naive, central, and effector memory subtypes. In these plots, high parameter data is reduced into 2 dimensions. Each dot represents a single cell, and cells or events that are close together on a T-SNE plot are similar to one another across dimensional space. For this CD4 T cell analysis, naive cells are in red, effector memory in green, and central memory in orange. Each population shows distinct clustering together, showing their similarities. The histograms on the right show the different expression profiles for checkpoint inhibitors, activation markers, and cytokine production. As expected, the biggest differences in expression patterns can be seen in CCR7 and CD45RA, with slightly less difference within the CD25 expression. A similar analysis of CD8 T cells was also performed to compare naive in red, effector memory in green, and central memory in orange. Naive and memory cells clustered in distinct populations, with central and effector memory more closely associated together than with the naive population. Again, as expected, CCR7 and CD45RA expression were the most altered when compared against all 3 groups. T-SNE analysis was then used to compare T cells stimulated in the presence or absence of the PD-1 inhibitor nivolumab. Analysis of CD4 T cells treated with the PD-1 inhibitor showed fewer cells expressing TNF and IL-2 than compared to those that were stimulated without the inhibitor. CD8 T cells stimulated in the absence of the PD-1 inhibitor had an increased TNF production when compared to cells that had been treated in the presence of the inhibitor. As expected, all groups showed an increase in TIM3 expression regardless of the presence of the PD-1 inhibitor. By day 13 within the CD4 T cell compartment, expression of PD-1 was still increased in the presence of PD-1 blockade. No difference in expression was observed for checkpoint inhibitors LAG3, TIM3, or CTLA4. The cytokine expression of interferon gamma, TNF, and IL-2 were similar between groups treated with and without the PD-1 inhibitor. Finally, T-SNE analysis was performed on the CD8 T cells following 13 days of stimulation in the presence and absence of the PD-1 inhibitor nivolumab. No difference in checkpoint inhibitor expression, cytokine production, or activation marker expression were observed when comparing treatment versus non-treatment groups. In summary, we used an in vitro model of T cell exhaustion in lieu of clinical samples. Chronic CD3, CD28 stimulation of pan-T cells resulted in changes to naive and memory T cell populations, as well as altered expression of checkpoint inhibitor molecules. Nivolumab treatment resulted in altered T cell subpopulations, and within the CD4 and CD8 T cell central memory, there were differences in PD-1, TIM3, and cytokine expression. Using the ZE5 to analyze a large multicolor flow cytometry panel allowed thorough analysis of multiple checkpoint inhibitors, activation markers, and cytokine production with limited sample. This is especially important in clinical cases where a doctor may need to obtain a maximum amount of information concerning a patient's immune status with a small amount of blood. Although we used an in vitro model, our 16-color panel could be used for immune response monitoring in clinical cancer research or infectious disease treatments. At this time, I'd like to take this opportunity to thank Chris Brampton, Liz Jordan, Michelle Scott, and Kristen Lynn for their work on this project. And thank you for listening to our webinar today. At this time, we'll take any questions.
And thanks. I want to remind everyone you can submit your questions. We will get to as many of them as we can. Thank you so much, Angie, for that really interesting presentation. Our first question comes from Mahesh, who wants to know, which software or bioinformatic tools can I use for FACS data analysis?
Hi, Mesh. We actually used Flojo to do our data analysis for this panel. And at that point in time, it handled all of our colors very well.
Terrific. Thanks for that. If you have a follow-up, go ahead and send that in. This one comes from Tatiana, who is more of a statement than a question. She says, I think it's not correct to identify TREG as CD127 low, CD25 plus, especially when you stimulated cells for 4 and 13 days.
Thank you, Tatiana. Actually, we only measured the Tregs at the 0 time point, and I would agree with you that at day 14 and day 13 following stimulation, we cannot use those markers to identify the Treg population. And while our panel does not include additional Treg markers, we just felt it was necessary to measure them at the beginning of our experiment.
Great. Thanks for that. Keep the questions coming. We'll get to as many of them as we can. Once we do not have time for, we'll receive a written response after our event today. This one is from Stephen, who wants to know, how long did it take to set up your compensation?
Actually, the Everest software was able to set a manual compensation very quickly, and it was identical to what we also checked in Flojo. On occasion, we have had to manually tweak 1 or 2 parameters, but for the most part, it doesn't take very long at all.
Okay, terrific. Thanks for that. Karen would like to know, what platform did you use for T-SNE analysis?
We used Flojo 10.4.2.
Okay, terrific. Thank you for that. This question comes from Evan, who wants to know, with your 16-color panel on the ZE5, are you able to add any more markers for, say, CD445R0 or CD27, or does this panel max out the ZE5 capability?
This panel does not actually max out the ZE5 capabilities. With the 5 laser instrument, we have 27 fluorescent parameters as well as 3 scatter parameters, and so we would be definitely able to add additional markers with that because we're only using 16 of the 27 fluorescent parameters.
Terrific. Thanks for that. Harris wants to know, he got here late, Harris, shame on you, and wants to know if it'll be available online. Yes. Starting tomorrow, you'll be able to see this event online. Just use the exact same link that you used to get here today, and you'll be able to share it with your colleagues. So thanks for asking, Harris. Another question from Tatiana. She wants to know, do you expect to see the same changes in tumor cells?
I think I would expect actually to see, if we were using clinical samples and using immune cells from a tumor model, I would expect to see hopefully more dramatic changes depending on the type of cancer model that we were using.
Okay. A lot of people saying thanks for your presentation, by the way. I just want to share that with you.
Thank you.
This question from Stephen, who says, did you use isotype antibodies or FMO to set your positive versus negative staining?
We did not use isotype controls. We actually used, this is a fully validated panel, so every FMO was used to set our gating.
Okay, terrific. Thank you for that. Keep the questions coming. We have time for a couple more. We'd love to get them to you. Here's another question. Just thank you for your nice presentation. A lot of questions. Another question on someone wants to know, what program did you use for the T-SNE analysis?
We used Flojo for our analysis, for both the traditional analysis as well as the T-SNE analysis.
Terrific. Thanks for that. Another question, this one from Sigrid, who wants to know, it's a long question, so give me a second. If you would analyze T cells in the context of a clinical trial with an immune checkpoint inhibitor, but you have to work with frozen PBMC, would you do the analysis immediately after thawing, or would you first incubate or stimulate the cells?
I would, if possible, generally do the analysis directly after thawing. But there are definitely reasons for waiting for stimulation or for recovery overnight. But I think to make sure that we have the most representative sample of what was frozen down, I would use it directly.
Okay. Kevin wants to know how many samples there were and how long did it take? Did it block during analysis?
We actually ran. This was an N of 1 as a representative to show that we can do this panel, and it took us roughly 2 weeks to do.
Terrific. That's all we have time for right now. I want to thank everyone for submitting your questions. We will get written response to them after the event. And if you joined us late or want to share this with a colleague, we've recorded this webinar, as I said, so you can use the same link that you used to attend live to view the event recording in the future. Again, my thanks to Angie Green for sharing her experiences with us. And for Bio-Rad, this is Michael Krieger saying thanks for joining.